Hey families, and welcome to Class 7. Class 7 is going to be a little different. It's all about Charles Lyell. So we're going to kind of take a step back and look at what we've done and kind of revel in what we've accomplished so far. And we're going to do that for two reasons. One is because we're about halfway through the course. It's a good time to sit back and reflect. And the second is because Lyell is the person who gave us time as a variable. So we're going to kind of see how that affects our thinking and our approach to biology going forward. So Charles Lyell, I couldn't find a good children's book about him. Or even a biography I thought was interesting. Not that I did an extensive search, but I did read through some of his own writing. And I will link to that because it's all available on the Internet Archives. So if you want to take a look, I mean, this guy really loved what he did. So it's fun to read what he wrote and just marvel at his illustrations. They're beautiful. But for now, I'm going to introduce Charles Lyell with the timeline of biology, the study of life, and just kind of walk through what we've learned so far. And then I'm going to introduce Lyell's research and what he figured out with his computations. And then we're going to apply that to some of our own work up to this point. So just to remind you, right, we started this course with Aristotle. As far as we know, he was the first biologist, and he was probably doing this work around 350 BC. And here I want to add in some art that I think is representative of the time we're talking about. I think it's just helpful to kind of get like a historical, cultural, zeitgeist kind of perspective what was going on at this time. So this is a statue of Apollo, and man, these artists knew what they were doing back then. I have no idea how someone makes marble look like folded cloth, but they sure did. And you could tell they just thought the human form was beautiful. So that'll give you an idea of what the times were like back then. And as for us, we sort of recapitulate this time by starting to make our own observations with the herbarium, the 6 samples that you got and the samples that you preserved for yourself. Now, skipping ahead about 750 years, we get into the medieval period. Remember, there was Pliny, there were the herbalists or the herb gatherers, and the physiologic. And that lasted for about 1,000 years. So at the beginning, around 400 AD, this is actually a mosaic that, or not a mosaic, a mural that I saw when I visited Rome. It's in the Church of San Clemens. So there's a church at street level, and because Rome is built on top of Rome, you walk down a set of stairs, and you find yourself in a Christian church from around 400 AD, and then you walk down another set of stairs and you're in a Roman home from like 90 BC. But this is a mural I've actually seen. So you can kind of get a sense of the art from 400 AD when the medieval period started. And remember that we got a couple more samples for our herbarium, which we ultimately had to set aside because they sort of went backwards in terms of biological knowledge. This is art from the end of the medieval period, around 1400 AD. And again, you can kind of see how far they came in 1,000 years of art. And that's really all we've got to say about biology at the time. Now, about 100 years later, things have really changed. And there's a lot to be said about why they change. But this is Michelangelo's David, right? The story David and Goliath. He's holding a rock. I've also seen this in Florence, and I cried when I saw it. It was so beautiful. And again, you could tell that Michelangelo just thought the human form was glorious. I mean, you could even see his ribs as he's kind of taking in a breath and assessing his situation. So what was going on in biology? Well, Vesalius was finally dissecting cadavers and wrote on the fabric of the human body, which was published in 1543. And, about 80 years later, Harvey figured out that blood circulated, and he proved it, through all these different means. And again, you look at these illustrations of Vesalius and Harvey, and you can tell that they revered the human form. Now, fast forward about 30 years. Oh, and don't forget, what we accomplish, right? We build all these heart models to understand how blood circulates through annelids and fish and mammals. So, fast forward about 30 years, you can see that line right there is not that far away from what Harvey was up to. What's going on? Well, in terms of art, we've got this Vermeer. It's called The Geographer, and it's so beautiful and so detailed. And what I love about it particularly is that this geographer isn't just focused on his protractor and his compass and his maps. He's looking out at the world. You know what I mean? He's not looking up at the heavens. He's not just looking down at his ideas. He's actively interested in the world. And as an intellectual, he's immortalized in this painting. He's revered, and I really like that. So, what's going on? Malpighi dissects a frog as it's still alive and actually sees capillaries. Like, this was the final mystery that Harvey was looking for. Like, what connects veins and arteries? Malpighi finds it. Swammerdam publishes his Historia Insectorum Generalis in 1669. And of course, Leeuwenhoek starts his correspondence with the Royal Society of London in 1673. And again, I just think these illustrations of his little animalcules is amazing. And we built all different geometries of lenses, and now we're using them to do further science, to gain more observations that we couldn't get otherwise. Now, at the same time, class 5, Reddy in 1668 is doing his experiments with insects, right? He's doing the first controlled experiment in biology. And so did we. We did pretty much the exact same experiment with our flies in the jar. Now, fast forward about 60 years, and you're getting these beautiful still lifes. And again, you can tell that the artist just thought living things were so beautiful and worth immortalizing in paint. At the same time, Linnaeus was starting to put together these correlations between related species. And again, you can see from these illustrations that he put in so much detail and attention to living things. What were we doing? Well, now we have a whole bunch more samples for our herbarium. We can start to make correlations to which species resemble each other and what makes them different from other groups of species. Now, if we fast forward another 100 years, this is a painting of Benjamin Franklin and his very famous key and lightning experiment. I would love to just go into that whole experiment, but we'll have to save that for physics. But in any case, what I love about this painting is that, again, he's revered for his scientific work. He's not portrayed the way a lot of scientists are in movies and television today, as crazy and sus, right? Like, he was really treasured for this work. So what's going on in the 1830s? Well, we mentioned Cuvier at the same time that we mentioned Linnaeus, although they were about 100 years apart, because, again, he's looking at correlations in fossils of animals, right? And starting to, piece together how these fossils are related. And Charles Lyell publishes Principles of Geology in 1830. And these are some of the illustrations. There's so many illustrations in this book, but these are just some of the illustrations of different mollusks. He knew tons about different species, even though principally Haha, he was a geologist. So, yeah, good on us. This is incredible. So let's move forward. Okay, so what did Lyell do? First of all, he had so many adventures and he collected hundreds of fossils and rock samples from 4 different continents, all over the place. In the course of his adventures and his sampling, he noticed a lot of really surprising things. For example, he had such a comprehensive knowledge of which fossils came from marine environments versus freshwater environments. And he noticed a lot of places where he found marine fossils in the middle of a continent. And I'm just pointing out a few of those places here. And likewise, he'd find freshwater. I mean, you could say that maybe the freshwater fossils on islands or near the sea or in cliffs might have come from a lake, but still, there were a lot of sort of surprising finds, these different sites. And then he did a very straightforward calculation. He was not the only one. So a number of his colleagues had done very detailed measures on how much sediment is moved across huge rivers like the Ganges and the Mississippi, and presumably all other rivers it deposit at the same rate relative to the volume of water that they move. So, for example, the Ganges deposits about 6 billion cubic feet per year of mud, and it drains a huge area of 400,000 square miles, which amounts to about 0.006 inch layer of mud across that entire surface area. And similarly with the Mississippi, another huge river in a different continent, there's about 4 billion cubic feet per year of mud that's deposited by the Mississippi. It drains about 100,000 square miles of the North American continent, which amounts to about 0.001 inch thick layer of mud over the entire area. So if we take those to be representative, and because they're so huge, probably are, and then take into account the surface area of the Earth's land, which is about 38 million square miles. The surface area of the water, all of our seas, is about 3 times that. So if all the land deposited evenly that's being deposited by these rivers is deposited evenly across the sea floors at a rate of between 0.001 inch thick layers and 0.006, then you have to, if you spread it out, you'd have to divide that by 3, because there's about 3 times as much area under the oceans as there is above the oceans. You get about 0.001 inches of deposition per year on the seafloor. Now, why does this matter? Well, Lyell didn't just take fossil samples, he also took a bunch of drawings and rock samples of a geological nature. So this is, again, one of the illustrations in his books. So he's looking at these different layers of deposition that have formed this rock, and so each layer is called stratum. So all 5 strata all together in this particular sample. Where is it from? Something hill. I forget where it was. I think it was somewhere in the British Isles, but I can't remember. In any case, all 5 of these strata together are about 20 feet thick. So if you're assuming from the previous calculation that the deposition rate was about 0.004 inches per year, it would take nearly 3,000 years for one of these 5 rock layers to be deposited. Now, At the time, I think it was still a pretty popular notion that the Earth was only about 4,000, maybe 6,000 years old. And that was coming from like the number of generations enumerated in the Bible. So the fact that you've got one bit of rock that's 20 feet thick, and that would have taken about 15,000 years to deposit. Like, that was a really surprising result, but it was kind of hard to argue with the math. Now, if you look at places like the Grand Canyon and all its strata, you start to think that maybe the Earth is really old. So Lyell goes into all kind of detail about different kinds of rock and different kinds of deposition and different kind of minerals and different kinds of fossils. I mean, this guy loved what he did and he knew his stuff. But really, he gave us this idea that the Earth could be very old, which means, it was really, I imagine, troubling to a lot of people that you see fossils that look very much like species that existed in the day, but clearly are no longer around. And how do you explain that? Well, if there's just this gradual change, that's really hard to explain if you've only got 4,000 to 6,000 years of Earth's existence to work with. But if you've got hundreds of thousands, millions of years, maybe, and billions of years, now you're talking. Now you can really start to imagine how things might have arisen and gone extinct. So in that respect, Lyell really gave us time as a variable. So what does this mean for our herbarium samples? Well, first of all, I'm gonna stretch out our timeline to include about over 500 million years. So in terms of the history of biology that we were just looking at, it does not even fit into that 1 pixel thick line at the very end of our timeline here. So good on humans for figuring so much out in such a short amount of time. But now we can really look at our herbarium samples, and you will notice there are some that are all in water. They're not necessarily little single-celled germs, although they often are. So these 3 samples are algae, and when we look at them with our questions, right? Do they live on land? No. Do they have roots and stems? No. Do they have 1 vein in leaves or many? Well, they don't have leaves, so no. Do they live in shady, damp, low-lying areas on land? No. Do they get big? No. Do they have seeds? No. Do they ever grow bigger than a person? No. Do they have flowers? No. Do they have fruit? No. So what if I told you that Lyell found, well, not necessarily, but that geologists have found fossils of algae more than 500,000 years ago, but not of any of the other plant types in your herbarium? Now, put yourself in the position of, pretend you're an algae, for example, right? Everything you need is kind of all in the same place, right? Especially if you're near the surface of the water. You've got sunlight, which allows you to make all the food you need. You've got air, which again allows you to breathe and break down your food and get energy from it. And you've got water, which helps with all of that and also allows you to propagate. Now imagine that you're trying to colonize this brand new space that's wide open. You can be a homesteader, but all your food and all of your energy and the place where you propagate are all in different places. How would you deal with that? So suppose I tell you that we do find the mosses, the bryophytes, 5 million years ago, but not before, or we find their fossils. Now, let's answer those questions. Do they live on land? They do, but they are not that complicated. They don't have roots or stems. They don't have leaves, so they can only get a little bit of that sunlight energy and a little bit of the nutrients from the soil. And they don't have seeds, so their offspring need water to propagate, so they have to kind of remain in those wet, low-lying areas. They certainly don't make fruits or flowers, or, like I said, not that complicated. But the fact that they do have some specialized tissue helps them kind of bring the nutrients from the soil and the water to propagate and the sunlight up above together in one place. So they're able to do what algae can't, which is colonize the land. Now, there's a big asterisk there. You do find algae on land. There's a special cheat. So one of the assignments, if you so choose, is to tell me what you think that big cheat is and where you find algae on land. Moving on. So now suppose I tell you that we find club mosses, the lycophytes. We find their fossils about 420 million years ago, but not later than that. So they look a lot like moss generally, although they tend to be a little bigger, a little more complicated. So they live on land and they kind of do have roots and stems. But they have leaves. It's just not very complicated. They don't get very big. They still need water to propagate because they don't have seeds. Their offspring are not protected from drying out. So again, they're kind of limited in their range. They don't get very big. Nevertheless, the fact that they have these more complex tissues so they can capture sunlight with their leaves and then use specialized tissue to move the sugars they make from the sunlight to the other parts of their bodies. And they've got roots to bring nutrients and water to these specialized tissues. That's a big deal. That's another sort of advance in dealing with the fact that on land, all of your nutrients, your energy, and your water are all in different places. So a little bit after that, you also find, around 400 million years ago, you also find fossils of ferns. And now we're getting a little bit more complicated vascularization. These are the vascular plants of the trachyphytes. Lycophytes, club moss, and ferns, polypodiopsida. I'm sorry, I'm not good at Latin. But again, you're starting to see bigger leaves, more complicated leaves with more complicated vascularization. But again, you usually see ferns in wet, low-lying places because that's what they need to propagate. But otherwise, they're specializing in getting, the sunlight and water and nutrients from different places and moving them around these specialized tissues. So, about 350 million years ago, now you start to see the gymnosperms. And the big advance here is seeds, right? So again, moss, club moss, ferns, they're all limited to really moist places because otherwise their offspring just dry out and don't make it, right? That's what they need to propagate. With seeds, the concern of drying out is much ameliorated. Your seeds can blow around in the wind and be just fine. In fact, for some gymnosperms, there are certain species of pine where it's helpful to have a wildfire to open up the cones and release the seeds. I mean, that's just unthinkable for ferns and club moss and regular moss. So that's a big advance. And then finally, for the first time, around 150 million years ago, that's when you first start to see the flowering plants, the angiosperms. So they don't all necessarily fruit, but they do have very well protected seeds that are produced from flowers, and that's where all the fruit plants are. Right? You don't get fruit as we understand it, really, from gymnosperms or ferns or club moss or moss. That you get them from the flowering plants, and you will notice with your samples, it's like most of your samples are probably angiosperms, and most of the plants you see around you are probably flowering plants. So what do we make of this? The fact that algae seems to be the oldest kind of species, and then the moss and the club moss and the ferns. So this is your big assignment for this week, is how did these come about, right? Like, why are these gradual adaptations to going from living in water to living on land? Where are they coming from? Why do you not see moss at more than 500 million years ago, for example? So, in the comments, please write, of course, in full sentences, what you think is going on there. All right. And again, congratulations. We've come a long